I guess people are succumbing to spring fever more and more.
That's for sure.
Let me continue on with where we were. I don't think I have any announcements I need to make. Next week gets back to normal, so Monday Live Group meets on Monday, Wednesday Live Group meets on Wednesday. Oh, I do have an announcement. The optional dissection that I was talking about, I want to update things a little bit. I was mistaken, not that this is a real issue with anybody so far, but I was mistaken about when I would be offering that, and my Tuesday and Thursday afternoon labs I won't be doing an optional dissection in their sequence. So the only times that are available for optional dissection are May 4th at 10:10, meaning the Monday lab group in here, or May 7th at 10:10, meaning the Wednesday lab group in here, which is meeting on Thursday the 7th there in the last week, as well as Monday the 6th. So if you're interested in doing the optional dissection, you need to let me know by tomorrow so that I can make the order for specimens. And let me know when you can come. If you can come to both those times, please let me know. The 7th is a Thursday, but the school's treating that as a Wednesday. So if you have Thursday classes, well, obviously you have this class technically on Thursdays. But if you have a class before this on Thursdays, that won't be meeting on the 7th because it'll just be, it'll just be a Wednesday schedule. Confusing myself there. But anyways, if you can make both of those, let me know. For the people that do opt to do this, I want to try to get, if there's a very small group, everybody to meet on the same day. So if you want to do this and you can do it on either day, let me know. If you can only do it on one day and not the other, let me know. And I'll try to make a determination of which day is going to be the optional dissection, so that the most people that want to can take advantage of it. So again, I need to know that by tomorrow. Send me a text or an email saying that you're interested and what time you can do it. If you're not interested, feel free to let me know that too. But if I don't hear from you by tomorrow, I'll assume that means you're not interested. If you then decide later on that you had always meant to do this and you never got around to it, never got around to telling me, you won't necessarily be able to do this. Okay, I need to know by tomorrow so that I can make orders. And if you do decide that you want to do it, I consider that a commitment to doing it. So don't say, yeah, I kind of feel like it, and then not show up on that day, because I will have ordered a specimen for you, and I expect you to be there to use it. So any questions about that? Yeah, the only new thing is that I had said I thought I was going to be doing it on Tuesday or Thursday afternoon, but in fact I won't. So just those two times that are available. Yeah, I think that's all the announcements I had to mention. To pick up with where we were last time, I pulled up essentially this picture. I did just a Google image search to pull it up last time. I might not have gotten this precise specific file, but I got some copy of this picture from somewhere. This one, obviously, because it's a little bit crooked, somebody scanned out of a textbook and posted it, which just, nowadays you shouldn't have to do that. But let me just make sure I've got everybody here. Okay. So this is a nice summary of what's going on in producing urine It's a 3-step process. Key down at the bottom presents those 3 steps in 1, 2, and 3. If I used a slightly different version of this picture last time to show it to you, they might have been A, B, and C, but still it's the same idea. The first thing that happens is filtration. The blood passes through the glomerulus, which is this bundle of capillaries right here, and everything gets filtered out of the blood except for the blood cells, large proteins, and at least larger lipids. They just can't pass through the filtration membrane. But everything else comes out. Water molecules, glucose molecules, amino acids, meaning nutrient molecules, and any other small molecule that might be present. And then the way that this picture is set up, instead of showing you the convoluted tubules, the loop, and all that stuff, this one long tube here represents the rest of the nephron. And the reason why it's so schematized here is because what happens in the rest of the nephron varies depending on exactly what we're talking about. But across the rest of the nephron, the other 2 things take place. First, number 2 is reabsorption, and 3 is secretion. Reabsorption means that some things that have been filtered out of the blood get reclaimed and moved back into the blood from the filtrate. We filter out nutrient molecules like glucose and amino acids, but we don't want to get rid of those. We want to keep those in the blood, so they move back into the blood. If there's a problem like diabetes, and so blood glucose levels are high, not all of the glucose will get reclaimed or reabsorbed because it's only going to reabsorb sort of the standard levels of glucose. And therefore, glucose will end up in the urine, which is diagnostic of diabetes. That, if you have high blood glucose levels, there's going to be evidence of that because glucose is going to be in the, end up in the urine, because not all of it will get reabsorbed. The reabsorption process here is to take in what's expected and required back into the body. And then secretion here is where things that are in the blood are moved into the filtrate, which is then going to be urine, as needed. It kind of looks like there's these 3 steps. Everything's filtered out, some of it gets back in the blood, and then some of it gets back into the filtrate. But really, step 3 isn't just putting things back into the filtrate. It's where a lot of the homeostatic mechanisms take place. So if we need to regulate sodium levels or phosphate levels or bicarbonate levels, pH, fluid levels in general, meaning water, potassium, calcium, all those sorts of things are regulated at really between reabsorption and secretion. If we need to lower our sodium levels in our blood, then we secrete the sodium back into the filtrate. If we need to increase our sodium levels in the blood, then we reabsorb it and keep it in the blood and don't get rid of it again. So these 2 processes help with a lot of the homeostatic mechanisms regulating the concentration of various things in the blood. Okay. What's not going on here is touching any of the real waste molecules that are being filtered out here. I don't remember if I said it this way for you or in lab at all, but just to be sure that I get this idea across. Imagine if you took aspirin this morning because you woke up with a headache or something. The active molecule in aspirin has its effects to reduce pain or swelling. Once it's done that, you don't need it anymore. Your liver is actually going to go through and modify that molecule a little bit, enzymatically change it so that it becomes essentially inactive, but it's still in your body, and so it's going through your blood. that molecule has to be filtered out of the blood. That's what's happening in step 1 here. You don't know at a physiological level that you have that molecule present in your body. You don't have some receptor showing the presence of that sort of drug-related molecule. And so everything gets filtered out. You're not going to reabsorb that material from the filtrate back into the blood because reabsorption is only going to be things that we know we want to get back into the blood. And you, again, at the physiological level, don't know about the presence of that molecule in your system, so you're not going to reabsorb it. So the filtration gets rid of everything, including all the things that might be in your body that you don't know about. I use aspirin as an example, but just as easily, it could be you've eaten some food that contains molecules that you absorb in your digestive tract, but don't really have any nutritional value. They're not necessarily macronutrients or micronutrients. So you don't need to use them. They might be toxic molecules, not toxic in the sense that it's going to make you sick because you ate it. It might just be something that's naturally in plants that you absorb, but you don't use. And so you need to get rid of that stuff too. So the process gets rid of everything that might be in your body with the filtration, and then reabsorption and secretion is where we can alter what's going on there, giving the kidneys the ability to manage a lot of the homeostatic mechanisms having to do with fluid balance, that sort of thing. So what I want to do is to talk a little bit more about what's driving filtration, and then talk about what's happening in reabsorption and secretion here, looking at those functions. So this picture, just to remind you, the picture I was just showing you isn't out of the book. I pulled that off of the internet. This picture is in the book, and what it's talking about is what's driving filtration. In the book, in this section, it's talking about net filtration pressure, okay, what's moving things from the blood into the filtrate. There's actually a few forces at play here, represented by these arrows. So the first arrow represents the force that is the blood pressure. Now this is called blood hydrostatic pressure. We don't usually call it that, we just call it blood pressure, but blood pressure is a hydrostatic pressure, which means the fluid, the water-based solution of our blood, is pushing against the walls of its container, the blood vessels. And then there should be an equal and opposite force back of the blood pressure, blood vessel walls on the blood. But when we have a leaky capillary like we have here, a fenestrated capillary, the blood will push through the wall of its container. And so the pressure at that stage is 55 millimeters of mercury. Going into your average capillary, which we saw back when we were talking about blood vessels, the blood pressure should be about 35 millimeters of mercury. So these capillaries are unusual. They have a higher blood pressure in those. And part of what helps maintain the structure of the capillaries, which are not used to such a high pressure, are the podocytes that surround them, which I showed you last time. So between the podocytes and the capillary walls, they can withstand the 75 millimeters worth of mercury, and that's what's pushing the stuff out of the blood and into the filtrate. But it has to work against a couple of opposing forces. The first one is, well, actually I'm going to talk about the bottom arrow here first, because it's another hydrostatic pressure. This is the capsular hydrostatic pressure. So as the filtrate fills the glomerular capsule here, it's going to be pushing against the walls of the container, so to speak, and the walls of the container are going to push back. And that's going to result in 15 millimeters worth of mercury, 15 millimeters of mercury worth of pressure pushing the filtrate back into the blood. Okay. It's not going to actually push the filtrate back in the blood because it's not enough to overcome the blood pressure that's at play there. But these are opposing hydrostatic forces, the blood hydrostatic pressure and the capsular hydrostatic pressure. Then the other one in here, which is not a hydrostatic pressure, this is osmotic pressure. The blood is a solution, suspension, however you want to describe it, with a certain concentration of stuff in it. And the filtrate is a solution with a certain concentration of things in it. There's more concentration, there's a more concentrated solution in the blood because of what doesn't get filtered out. The cells and the proteins and those sorts of things keep the blood more concentrated than the filtrate. And so osmotic pressure is going to naturally push water out of the filtrate back into the blood. The blood is hypertonic compared to the filtrate, which is hypotonic. And whenever you have a hypo and hypertonic solution separated by a membrane, osmotic pressure is going to result, and water is going to move from the hypotonic solution into the hypertonic solution. Hypertonic means that it's more concentrated. Hypotonic means that it's less concentrated. You could also think of hypotonic has more water in it than solutes, compared to a hypertonic solution, which has more solutes than water. Kind of an oversimplified way of putting it. But the fact that there's more water in the hypotonic solution, the water moves to try to dilute the hypertonic solution until the two are going to get to equilibrium. That's just a standard effect that we'd see across unequally concentrated solutions. And we have unequally concentrated solutions here because of what does not get filtered out of the blood. The amount of osmotic pressure pushing back is 30 millimeters of mercury. If you combine these three forces together, considering their different directions, 55 millimeters of mercury minus 30 millimeters of mercury minus 15 millimeters of mercury gives us a net pressure driving filtration of 10 millimeters of mercury. That's what we call the net filtration pressure, or NFP. Excuse me, NFP. In the book where this picture's presented, they basically show the math there. The 55 minus 30 minus 15 equals 10, okay, just in defining net filtration pressure. But that's what's driving it. So there's enough pressure behind this to get the filtration to take place. 10 millimeters of mercury is not a lot of pressure. Okay? It's enough to drive the filtration here. But if anything alters any of these values, then we've got a problem. And the most likely thing to change here is the blood hydrostatic pressure, or blood pressure. If your systemic blood pressure is low, then there can possibly be a problem between or within this calculation here. Now, the 55 millimeters of mercury is probably based on the average, say, of 120/80 blood pressure. That's not really the average. I don't actually know what the true average of blood pressure across the population is, but really the 120/80 is sort of an upper limit. That's a place to say above this means that the pressure is getting too high, systemically speaking. But let's say 120/80 pressure at the aorta, which is essentially where it is, or in the brachial artery, corresponds to a mean arterial pressure or blood pressure of 55 millimeters of mercury in the glomerulus. If your blood pressure drops to 110/70, or that's what it naturally is, that's actually a better blood pressure to have. It's easier in your cardiovascular system. But if we drop from 120/80 to 110/70, what do you think is going to happen to the blood pressure that we see here?
It's going to be less.
How much less? I just told you we're assuming that with 120/80, you'd see 55 millimeters of mercury here. If we go from 120/80 down to 110/70, how much of a drop are we going to see here? 10. Yeah. So we'd go down to 45 millimeters of mercury. 45 minus 30 minus 15 is 0. Okay. that little bit of change, if that was all that was at play here, would mean that the kidney could no longer filter out wastes from the blood, and that results in kidney failure. Now, working on what we consider sort of averages, the 120/80 corresponding to 55 millimeters of mercury here, is sort of where I started with that. That might not be entirely true. And in fact, there are mechanisms that help regulate this. So the glomerulus wants to be sure it keeps its blood pressure at 55 millimeters of mercury. And systemic changes in blood pressure will be overcome by changes that are happening in the kidney and then how the kidney affects blood pressure as a whole. After I talk about the process of filtration, reabsorption, and secretion stuff, I'll come back to the regulation aspect of what's happening here. So we'll come back to that idea of how the kidney keeps the blood pressure here at 55 millimeters of mercury. So moving on in the book, the next picture I want to talk about is just the idea of what's happening of, in reabsorption and secretion. So this is just showing the whole nephron and the collecting duct that it connects to. The arrows pointing out are referring to reabsorption, meaning that components of the filtrate are moving out to get into the blood. And then arrows pointing in are describing where things that are in the blood will move into the filtrate. Okay. So first off, I want to point out glucose and amino acids are at the top of this list. They're going to be reabsorbed immediately, okay, right in the proximal convoluted tubule. And then there are a number of other things that can be reabsorbed. I mentioned that proteins don't get filtered, but there are some proteins, if they're small enough, they will get filtered out. So those will get reclaimed too. Vitamins would be micronutrients. So they're going to get reclaimed because we want to use those. And then we have lactate, which is kind of another way of saying lactic acid. Urea and uric acid, which are not the same thing, but they both obviously have in their root, the root that gives us the word urine. And then a number of ions or electrolytes that we find in the system. Those can be reabsorbed. Now, it might seem strange that lactate, urea, and uric acid are being reabsorbed, but if you look down here, at least for the urea and uric acid, they're also being excreted. Okay. For urea, at least, there's so much of that in the blood. It's sort of the main waste product in the blood. It moves kind of freely in and out of everything. So there's concentration gradients that'll drive it out of the filtrate, but then we actively move it back into the filtrate to overcome that, which we see sort of there. If you look at the various ions here, a lot of them are repeated in other places because there's other movements in and out of those. So we have, say, potassium coming out here, potassium going in here. Let's see. Chloride, sodium, bicarbonate, et cetera, et cetera. Water's being taken out along several of the areas here. What would be kind of nice, and I've seen other books kind of present it this way, and I'm sure that this book didn't present it that way because it didn't want to copy the other books directly, but some of these arrows are conditional, optional, whatever you want to call it, which is to say that the things that are listed in the boxes here don't necessarily have to come out at those stages. For example, water that's shown coming out, down here. There we go. It's coming out here in the collecting duct. That's actually regulated quite a bit by a hormone when we need to retain water, when we're not taking in new water in our system. We do, in fact, move water out. But If you're getting plenty of water, then it's not going to be taken out of the tube at that point. It'll stay in the urine. But this is kind of an overview of where different aspects of reabsorption and secretion take place. This is looking directly at a cell in the proximal convoluted tubule, and it's showing several different processes that move these things across the apical membrane of the tubule cell, which means between the lumen where the filtrate is, and then eventually those things will move out across the basal surface of the cell and into the bloodstream. This is sort of a summary. This isn't necessarily happening in every single cell, but these are the kinds of things that can happen. A lot of it's driven by an energetic pumping of sodium out of the cell, and therefore potassium is going to come in. Energy is expended here at the sodium-potassium pump. But if we look, say, at this first one, sodium's coming in. And when sodium comes in, it helps to push hydrogen out, okay, hydrogen ions. Sodium's coming in down a concentration gradient. Because ATP is being used to pump sodium out, the concentration of sodium in the cell is going to be lower than the sodium that's going to be in the filtrate. So sodium wants to move into the cell, and what these cells do is they take advantage of that concentration gradient to pump other things out, like hydrogen. Okay. I want to point out, importantly, the glucose and amino acids here, they are pulled in with sodium. These are co-transporters. And actually, the proteins that do this are the identical proteins that are used in the digestive system to pull these nutrient molecules in from the lumen of your digestive tract. So again, ATP is used to build up a sodium concentration gradient, and as sodium comes into those cells, they bring along amino acids or glucose. The two processes are pretty much the same, okay? Moving nutrient molecules from the lumen of a tube across a cell into the bloodstream. And so there's no reason for the tubule cells to try to reinvent the wheel, so to speak. The mechanism already exists in the digestive system, and so the urinary system just uses the exact same thing. There's already a gene for that protein, that co-transporter, and so it just gets used again here. Now, the way I'm characterizing that is probably a little bit oversimplified or unfair or anthropomorphic or whatever. I said that it already exists in the digestive system, that the urinary system doesn't reinvent the wheel. Which came first isn't really important. It's just the idea that the body is going to be efficient about how it does things. And if the same process has to take place in two different systems, it might as well use all of the same players to do that. You would probably not feel too comfortable thinking about how your body interacts with what's going to be urine is the same way that your body interacts with what is food. We usually think we don't want to mix those two things together, but at the molecular level, the processes that move these things out of the lumen of the tube into the cell, which then sends it into the bloodstream, is identical. Okay, and some of that's similar in some of these other situations. These are just general cellular processes of moving these things across membranes. If it's happening somewhere else in the body also, it's going to be using the exact same process. Which is just another thing that ties two of these systems together within the environmental exchange aspect within this unit in the book. The digestive system and the urinary system actually work very similarly in moving nutrients and vitamins and minerals, et cetera, from the lumen of their tubule structures into the bloodstream. Okay, so it all just works in a very similar way there. And it's a lot of it's driven by building up a sodium concentration gradient with a sodium-potassium pump in the basal side of the cell, which we see there. Some things don't need that kind of help, like magnesium and water down here. They just move freely across this. So if you have low magnesium levels in the blood, the magnesium in the cell is going to be low, and so you'll reclaim the magnesium. However, if you have high magnesium levels in the blood, you'll be filtering some of that out. But if the concentration gradient isn't strong enough, the magnesium won't move. Okay? Trying to get things back to equilibrium. So if you have high blood magnesium levels, you'll filter that out and it won't come back in just because there's already plenty of magnesium there. It's not going to move back down. But if blood magnesium levels are low, then magnesium will just move across in a down a concentration gradient, just as an example. It's one of the things in this picture that doesn't have anything tied to it. The picture here for water just shows an arrow. Water does pretty much freely diffuse across membranes, which actually is counterintuitive, hopefully. Water is obviously hydrophilic. It's water. It loves itself. It's not a self-hating molecule or something like that. So being hydrophilic, it should not be willing to pass through the lipid bilayer of a membrane. Okay? That's true, but just in general for things crossing over lipid bilayers, it happens every so often, once in a million, once in a billion times, something like that. There's so much water that overall there is a net movement of water across the membrane of a cell that we can measure to some degree. So oftentimes in textbooks, you'll just see water moves across the membrane. This actually is not just free movement of water. It's just there's so much water that there will be a measurable net movement of water across membranes. There are some cells, not the proximal convoluted tubule cells, but other cells in the tubules of the nephron that are more exclusive. They keep water out, making it less likely for water to just move through. But then we have the ability to move it specifically through by opening up channels specifically for water. So we have some control over when water is moved from the filtrate into the blood. And I mentioned this a second ago, talking about the collecting tubules, collecting ducts. We regulate the movement of water from the filtrate into the blood, corresponding to whether we're keeping ourselves hydrated or not. And I'll get to the hormonal control of that a little later on, which might be coming really quickly. Oh, that's right. This is another picture in a proximal convoluted tubule cell. It actually shows some of what we just saw. So here's that sodium potassium, whoops, sorry. Here is a ATPase that pumps sodium out. This happens to be one that's also, oh, this is a little bit different. Pulling hydrogen in, here we see the same sort of thing. What's most important here is this is showing how hydrogen and bicarbonate are tied together again here with the carbonic acid molecule and the carbonic anhydrase reaction. That reaction takes place in the lumen of the tubules, so that hydrogen ions that are pulled out end up being transferred to water, and then carbon dioxide comes out. The carbon dioxide can then be sort of recycled through the same thing happening inside the cell. Seems kind of strange, the same things happening on both sides, but it's a matter of making use of the hydrogen ions and getting them through, and this produces bicarbonate, and the bicarbonate can move out of the cell and into the blood, where it can then again provide a buffer system for the blood. Okay, so that's the third place that we've seen bicarbonate being produced, which will eventually move into the blood. Okay. The way they have this depicted here, I don't know, I'm not familiar with this. I'm not a biochemist by any stretch of imagination. The bicarbonate here, they have plus sodium and pointing across. I was always under the impression that bicarbonate moved through the membrane in its own fashion, tied to chloride. It's entirely possible. I'm not aware of a sodium-dependent bicarbonate process. So I don't want to say this is wrong. I'm just not familiar with it. But it doesn't matter. When we saw bicarbonate and chloride tied together with the parietal cell in the stomach, that's important because the chloride's part of what the stomach needs for its hydrochloric acid. If chloride's not involved here, it's not earth-shattering or anything. It's entirely possible there's a different way for bicarbonate to move across. But it ends up in the blood where it can act as a buffer here also. And also, as it's buffering the blood, what's happening here, moving these hydrogen ions across the cell, both the basal and the apical surfaces, by pulling hydrogen out, we're also helping to control blood pH. So one of the things that the kidney is responsible for is regulating blood pH, both by adding buffer to the system, which is sort of a minor part of it, and more directly pulling hydrogen ions out if blood pH is too low. Okay, so it'll get out there. I want to point out in the loop of the nephron loop here, there's sort of a very specific thing going on. And this gets back to the idea of regulating water. Now, the loop descends down into the medullary pyramid, okay? So the border between the cortex and the medulla is probably right here somewhere. And so most of what's happening, especially in the convoluted tubules and the glomerulus, is happening in the cortex. But the point of the loop is that it descends down into the medullary pyramid. And the medullary pyramid is interesting tissue. It does something that we don't tend to see, which is it maintains a range of concentrations. The concentration of the interstitial fluid is measured in this unit, milliosmoles per kilogram. The standard value for that, what we're going to find for pretty much any interstitial fluid in the body except for right here, is about 300, maybe a little less. But as we go deeper into the medullary pyramids towards their tip, where the urine is going to be excreted, the concentration in the of the interstitial fluid in the tissue gets more and more hypertonic. So what's happening with the loop is as it's going down, the filtrate is passing through this tissue, the more and more hypertonic tissue solutions. And so the concentration that we find in the filtrate increases. Water comes out of the filtrate as it's going down in the thinner part of the loop. Water comes out, which makes the filtrate more and more concentrated as it gets deeper and deeper in here. And then as it goes back up, water is not allowed across the wall of this part of the tubule. So this is one of the places where water doesn't just happen to cross over cell membranes. It's not allowed. The cell membranes are more impermeable to water than your average cell membrane. So water stays in this, which a lot of water has been taken out to concentrate the urine down. But as it goes back up through these ascending interstitial concentrations, the solution has to also become more and more dilute. And it does that by removing solutes, primarily, actually I should say exclusively, sodium and chloride. So in the filtrate, there's a whole bunch of stuff dissolved in there, giving it an overall concentration, which is in this unit that we're talking about here. as it goes up the loop of Henle out of the medullary pyramid, it's going to become diluted. The water is sort of stuck in its position. No new water is going to come in to dilute that. Instead, it's going to be diluted because some of the things dissolved in it move out, specifically sodium and chloride. When we get back up here, the concentration of the filtrate is low again. And actually, the process makes it, can make it very, very dilute. So it gets down to maybe 100 milliosmoles per kilogram compared to the 300 that's actually in the tissue. But it's using this change in concentration as we go back up to drive moving the sodium chloride out, making a dilute filtrate, but it's concentrated with the waste molecules we want to get rid of. What's really diluted out of it are things that we have control over, like sodium and chloride. And then that filtrate that's dilute overall, but concentrated with the waste products we want to get rid of, moves into the distal convoluted tubule, where it eventually goes to the collecting duct. So let me go back to, whoops, this picture here. Okay. So as we go through the loop, what I was just talking about here, the water's pulled out as it gets concentrated, going deeper and deeper in here. And then as the filtrate goes back up, it gets dilute because sodium and chloride especially are pulled out. When it gets to the distal convoluted tubules, at this point, it's dilute overall, but it's had specific solutes removed to get it to that dilute state, and other solutes are left in there, the ones that we want to eliminate as urine. As it leaves the distal convoluted tubule, a couple of more things can happen to tweak the amount of things that are in there. And then it moves into the collecting duct. At this point, it's considered urine. Once it leaves the nephron, it goes in the collecting duct. It's nephron. It's urine. Going through the collecting duct, water can enter or exit, really, the filtrate, which might help concentrate the urine as it goes back down. That happens under the control of a particular hormone called antidiuretic hormone. Did I define that hormone last time? So I'll just remind you, whoops, of that. Antidiuretic hormone, which is abbreviated ADH. Didn't write any of this up on the board. Okay, so some of you saw me write this up on the board, but I'm going to write it up on the board again just to be sure. Okay. The diuretic is an adjective, maybe, but the noun form of this word is diuresis. Uresis means to urinate. Diuresis means to pass water in the urine. Antidiuresis is to retain water so you don't lose water in the urine. So this hormone basically causes urine to be retained. Causes water to be retained and not lost through urine. This hormone is is produced by the hypothalamus and released at the posterior pituitary. It controls the expression of a protein called aquaporin, which is a water channel. without ADH present in the system, if you're not releasing that, the water will end up in the urine. With it, with ADH present and aquaporin being expressed, water will leave the urine and go into the bloodstream to keep you from becoming dehydrated. ADH is most often released, or is mainly released overnight. Why would that be necessary? What's special about overnight in regards to water? Yeah, because you're asleep, you're not taking in water. During the day, when you have the ability to take in water, it doesn't tend to be released. However, if you aren't taking in water for some reason, ADH levels might go up to help regulate that. Without the water in the urine becomes more and more concentrated, and you'll see that in the urine as it's a darker and darker shade of yellow. Okay. Now, there's actually two types of nephrons, which the book might mention in the text, but they don't have a representation of this in the picture of the nephron that we looked at last time. There's what are called cortical nephrons and juxta medullary nephrons. Medullary nephrons. I left a little space there because I'm going to add a few things in describing these two. What we've talked about nephrons so far applies to both of them. I just want to highlight the differences and what that means functionally between them. The term juxtamedullary means these are next to the medullary border, okay. Specifically the glomerulus. So let me go back to the book for a second. It's not in this section, sorry. We're just talking about the nephron. There we go. The interlobular vein, as the book calls it, it's what I'm calling the cortical radiate vein. It's branching off of the arcuate artery, the interlobular artery, or the cortical radiate artery is branching off the arcuate artery, which is going right here. It's basically the marker of the border between the cortex and the medulla. The loop of the nephron extends past that border into the medullary pyramid. The location of the glomerulus is where the name comes from. So a juxtamedullary nephron, its glomerulus would actually be down here, closer to the border with the medulla. It's the juxtamedullary next to the medulla. I said that first because really the comparable aspect of the cortical nephron is that the glomerulus is more superficial in the cortex. So there are a bunch of nephrons in a bunch of glomeruli in the cortex. The ones that are closest to the medullary border are the juxtamedullary ones. The ones that are not next to the border there are cortical. The majority are cortical. Okay. So they're sort of our standard nephron. The juxtamedullary ones are special. Cortical nephrons have a short nephron loop. And, oops, Juxtamedullary nephrons have a long nephron loop. Now, again, back to this picture I was just pointing out, the way they've drawn this, which is not showing the relationship between the loop and the medulla, it suggests that this is a fairly short loop here. Okay. But if we go to this picture here, there's nothing in the picture that suggests that it's a long medullary loop. All it is as we get deep into the medulla, medullary pyramid, we get down to these levels of concentration. A cortical nephron's only going to extend down a little ways, maybe to about where the 600 level is here. So a nephron that goes this deep into the medullary pyramid, down to where the concentration is at 1,200 milliosmoles per kilogram, that's just very, very deep in the medullary pyramid. Which brings us to the functional difference between these, which is juxtamedullary nephrons are specialized for concentrating urine. And the cortical nephrons handle regular urine production in more dilute urine. So during the day, when you're taking in water, a lot of that water will be passed on into the urine, and you'll get rid of it when you void your bladder. The cortical nephrons are doing that. But when you need to concentrate urine, the juxtamedullary nephrons are specialized for that. And they make use of that concentration setup in the medullary pyramid here that we see. Because they go so deep into the medullary pyramid, they get down to where it gets really concentrated. So the urine, the filtrate at this point, gets very concentrated and then it goes back up. The same process is happening in cortical nephrons. They just don't get as deep into this, so they don't concentrate the urine quite as much. This process, like in terms of the juxtamedullary nephrons, is about making concentrated urine, which is related to retaining water when you need that. That's not the only reason for this. This is also making sure that the filtrate, which is getting concentrated with the things we want to remove. So even if it's a cortical nephron that's only going part of the way down in here, it's still going to pull water out and then pull sodium chloride out. And what we get up here is overall a dilute solution, but it's got the waste products fairly well concentrated. It just from this point on through the distal convoluted tubule and the collecting duct, when ADH is present, the urine that's going to result from what comes out of this will end up getting concentrated again as the collecting duct goes back down through all these levels here. The collecting duct is running parallel to this, so it's going through these descending layers of more and more concentrated tissue also. So in the collecting duct, the urine can be manipulated based on water and aquaporins and ADH to make it concentrated or more concentrated, less concentrated. The juxtamedullary nephrons are supplying the filtrate that's best concentrated with those waste products to get rid of them. And then in the collecting duct, in the collecting duct, the last of that concentration takes place, helping you to retain the water that you need as it's coming back down. So, two types of nephrons there. There's one other thing about the juxtamedullary nephrons, which I don't think the book mentions, but I want to just point it out in case it comes up somewhere. The peritubular capillaries form what's called the vasa recta around the nephron loop. To write something like that under the cortical nephron would just be to say that there's no vasa recta there. Again, I don't think the book has a good representation of this. It might describe it in the text, but it doesn't represent it in the picture. Here's a picture showing the peritubular capillaries, or the capillaries that are around the nephron, including around the loop. In a juxtamedullary nephron with a very long loop, the peritubular capillaries sort of look like a rope ladder, and that's called the vasa recta. Around cortical nephron, they are not quite organized like that. And actually, now that I've pulled this up, there's a few other pictures here. This one is actually showing two nephrons in a little bit better relationship to where the border is. The cortex is what's outside or above in this picture, the arcuate artery and vein. And then the medullary pyramid is what's below that. So the glomerulus of this is further up in the cortex. The glomerulus of this is right next to the border. And that's the idea of the glomerulus location related to things. The peritubular capillaries here are just a mash of stuff all around this. For the juxtamedullary nephron, it's we have this rope ladder looking structure around the loop, which is called the vasa recta. This is a little odd. It doesn't show anything around the loop of the cortical nephron. It doesn't really show anything around the convoluted tubules of the juxtamedullary nephron. Not because they aren't there, but apparently they're just trying to point different things out. And it's kind of small, but maybe you can kind of see, here we have what's supposed to represent a lobule of the kidney. So we have the cortical nephron here and the juxtamedullary nephron a little bit lower down. It's not really much different from the one I just showed you. Now, that's probably the best one for oops, not that one. Where was I? It's kind of the same picture, but now it's got a much longer loop. But that kind of gives you the idea of the difference between the two. The position of the glomerulus, the length of the tube, the length of the loop, and then the vasa recta surrounding the loop of the juxtamedullary nephron. Those are the structural differences between them. And then functionally, the big difference is one specialized for making concentrated urine. The other one's just making your average, more dilute urine that's produced throughout the day. Any questions about this stuff before I move into the last thing? Yeah.
I do. So when we're more hydrated, the juxtamedullary nephrons usually work, but when we're less hydrated, the cortical nephrons work.
No, you actually got it backwards.
Okay,
The less hydrated state is when your juxtamedullary nephrons are going to be making dilute urine so that the water stays in your body to counteract that less hydrated state. And then when you're more hydrated, you can pass all the water you want to in urine, and so the cortical nephrons are there. The two types of nephrons are always working. It's just depending on the situation, one's going to be more important than the other. Okay. And it's not just the nephron that's doing that. The ADH thing I talked about is always also going to play into the hydration state. But the very dark urine that you'll see first thing in the morning, a lot of that's produced through the action of these juxtamedullary nephrons, helping to concentrate the urine like that. Okay, so I actually had the book here on the endocrine regulation thing. There's not any pictures in here. I just want to talk about a few things, and I'll just have the book up so you can see the names of these things that I'm talking about. There are a number of hormones that play a role in regulating what's going on with the kidney. Several of them are responsible for regulating blood pressure, and then there are other ones that are responsible for other aspects of things. The first set here, these are all hormones, but they're interrelated. They work as kind of as one. Renin, which I talked about before, is released by the juxta glomerular cells when filtration is not working up to snuff. Remember, the macula densa in the distal convoluted tubule is sampling the concentration of sodium and chloride and that sort of stuff in the filtrate. And if it's not at the right levels, then renin's released by the juxtaglomerular cells to help change things. Locally in the kidney, that's going to cause the afferent arterioles to dilate and the efferent arterioles to constrict. By letting more blood into and less blood out of the glomerulus, that's going to increase blood pressure in the glomerulus to keep it up at, to keep the net filtration pressure up so that filtration is taking place. But then renin also causes changes systemically. It causes the conversion of angiotensin. There are two angiotensins, angiotensin 1 and angiotensin 2. Renin causes angiotensin 1 levels to increase, and then angiotensin converting enzyme converts angiotensin 1 into angiotensin 2. Angiotensin 2 is then a hormone that is a very potent vasoconstrictor, which is going to increase systemic blood pressure. Okay, so back to what I was talking about with filtration pressure earlier. If your systemic blood pressure is low, even 120 over 80 dropping to 110 over 70, that might represent a 10 millimeter of mercury drop in the blood pressure in the glomerulus, which would basically cancel out filtration pressure. And so as those kinds of changes can have a profound effect on the kidney can have a profound effect on systemic blood pressure. And angiotensin 2 will cause vasoconstriction systemically, so all of your blood vessels constrict, and therefore your systemic blood pressure goes up. And as systemic blood pressure goes up, the filtration pressure at the glomerulus is going to go up too. Okay. Renin and angiotensin together are usually considered one thing. Renin is just a hormone to stimulate the conversion of angiotensin 1 to angiotensin 2. Stimulate the production of angiotensin 1 that can then be converted into angiotensin 2. And then angiotensin 2 is what actually does the work in vasoconstriction. Angiotensin 2 will also increase the production of aldosterone. Okay. So angiotensin 2 and aldosterone are both going to be present in the bloodstream at the same time. Ultimately, because of renin increase from the kidney. But angiotensin 2 has its own things to do, and aldosterone has its own things to do. The book highlights the vasoconstriction aspect of angiotensin 2, and then aldosterone, it highlights that it helps regulate sodium levels. High aldosterone in the blood is going to cause the kidney to excrete sodium, okay? So, they don't actually have an arrow pointing to sodium, but sodium, all the arrows that have sodium next to them, the sodium's coming out of the filtrate. What aldosterone is going to cause happen is for sodium to be pushed back into the filtrate. Now, the sodium's not the important part. The sodium's what it's acting on, but when sodium leaves the tissue or the blood, which are essentially synonymous, and goes into the filtrate, that's going to make the filtrate hypertonic compared to the blood in the tissue, which would then be, relatively speaking, hypotonic. And so water in the blood will move into the filtrate to dilute it back to an equal concentration. So aldosterone's manipulating sodium levels, but it's doing that to manipulate water levels. Okay? Water moves back into the filtrate because of the sodium levels, and that's going to reduce the amount of water in your blood, which means that blood volume will go down. And as blood volume drops, blood pressure, wait, did I say that right? I'm getting that confused, sorry. Everything I just said is wrong. No, everything I just said is backwards, is all it is. It's not sodium being pulled into the filtrate, it's sodium coming out of the filtrate. Something else moves sodium the other direction, which I'll talk about in a second. But anyways, increasing sodium levels in the blood will increase water levels in the blood. Okay, so everything I just said was wrong, but only because it was the exact opposite of what I meant. Sodium is going back into the blood, increasing water levels in the blood. Okay. That's going to increase blood volume, and therefore blood pressure will go up. Okay. Angiotensin II increases blood pressure through vasoconstriction. Aldosterone increases blood pressure by increasing blood volume by manipulating sodium levels. Okay. Most people are probably familiar with recommendation if somebody has heart problems that they should eat a low sodium diet. And the reason for that is you want sodium levels low because then blood volume levels will be low also. Aldosterone is going to counteract that. So if you eat a low sodium diet, aldosterone is going to get sodium levels in your blood back up if you need blood pressure to go up. But usually if you have cardiac issues and you're eating a low sodium diet, your systemic blood pressure is probably already high. But in a normal situation, aldosterone is about moving salt from the filtrate into the blood. Okay. Sorry, I started getting that backwards. I got ahead of myself. So that's what angiotensin and aldosterone are essentially doing. And these three are usually thought of as one big thing because renin gets everything started. Renin increases angiotensin levels, and angiotensin II causes aldosterone levels to go up. But really, angiotensin II and aldosterone are two separate hormones that are causing changes in blood pressure and blood volume. Then I've already talked about antidiuretic hormone, but we can consider what that has to do with blood pressure also. I've talked about it in terms of hydration levels, but if you're retaining water, then blood volume is going to go up and blood pressure is going to go also. Okay. Aldosterone, as I just described it, it affects sodium levels, and through sodium levels ends up increasing the amount of water in the blood and therefore blood pressure. Antidiuretic hormone acts on the water directly, again, through that aquaporin channel. Okay. The book mentions endothelin. I'm not going to say anything about it just because this is the first book I've dealt with that actually deals with it, which means I don't have any questions that deal with endothelin. But it's one of the other hormones that play a role in here. And then here's what I was getting wrong, or this is why I was getting things wrong. There's another class of hormones called natriuretic hormones. Okay. Natriuresis, like diuresis, is talking about how something moves through the urine. So again, the uretic or uresis part of the word is talking about urine production. And instead of diuresis, this is natriuresis. And the Latin name for sodium is natrium. So natriuretic means passing sodium in the urine. Okay. So natriuretic hormones do what I was accidentally saying aldosterone does a second ago. They cause sodium to move from the blood into the filtrate. And in doing that, water will follow it. So aldosterone and antidiuretic hormone increase blood volume, mainly water. Natriuretic hormones decrease blood volume by pushing sodium out, and therefore the water goes with it. Now, the natriuretic hormones, there's multiple ones. The two main ones, oh, that's weird. are the book does something kind of weird, which has kind of bothered me. They refer to as ANH, which stands for Atrial Natriuretic Hormone. More commonly, I see this referred to as ANP, which is Atrial Natriuretic Peptide. Atrial is referring to the atrium of the heart, and that's where these hormones are released primarily. When your blood pressure, your blood volume is high, then the pressure on the wall of the atrium is going to be significant. And so stretch receptors in the atrial wall will release this hormone to get blood pressure and blood volume down so that the heart's not being overloaded with blood. And so that's what the A in atrial natriuretic hormone, or peptide as I prefer to call it, refers to. There's also a BNP. So here, book being very strange, they actually use the word peptide here. There's an ANP and a BNP. And in the book, they call it ANH and BNP. And they're saying B-type natriuretic peptide, which is fine. The B actually stands for something else. But I'm not going to talk about that because it'll just confuse things. But BNP is released by the atrium also, okay, in response to high blood pressure and volume. And that'll help lower things. Now this goes on to talk about parathyroid hormone, which I'll deal with later on. I want to stop here because what we're talking about here are hormones that regulate blood pressure. So again, we have renin-angiotensin, which basically causes vasoconstriction. Aldosterone, which causes sodium retention and therefore water retention. That's not how you spell retention. Tension. And ADH that directly causes water retention. All of those will increase blood pressure, okay? And then we have the natriuretic hormones, ANP and BNP, which cause sodium excretion and therefore water excretion. Just to highlight that. Now in the book, they put renin-angiotensin-aldosterone together because aldosterone is made, that's not aldo, sorry. Aldosterone is made because of angiotensin levels, but it has its own effects. The first three raise blood pressure, the last type lowers blood pressure. So they sort of act against each other. And between the two sets, we can have pretty fine control over blood pressure, which is all based on what the kidney is doing and how the heart responds to that. Okay. Actually, I'm not going to talk about, where'd it go? I keep missing it. Parathyroid hormone, but it is mentioned here. I've talked about parathyroid hormone before in this course. It came up originally in regards to calcium homeostasis in the bones in ANP1. It's repeated in the endocrine system chapter. So I'm not going to go through it again here. It's mentioned here just in what parathyroid hormone does with the kidneys as opposed to the bones directly. Okay. But leaving this out, I do want to stress it's not one of the blood pressure hormones. It has its parathyroid hormone is involved in calcium homeostasis, not blood pressure homeostasis. These ones that I went over are interrelated there. So that wraps that stuff up. If I think of anything else I want to cover, I'll start off with that next time, but we're going to be starting the reproductive system next. So